Hello and welcome to some biology class. Friends, today we shall discuss about the principle, procedure and applications of radioimmunoassay. Radioimmunoassay, also known as RIA, is a competitive assay technique in which the radio-labeled antigens compete with unlabeled antigens for binding with a limited amount of antibody to form antigen-antibody complex. At the end of the reaction, the antibody-bound antigens are separated from free antigens by a suitable method. The amount of labeled antigen-antibody complex is inversely related to the concentration of unlabeled antigen. A standard curve is then plotted by taking different known concentrations of unlabeled antigen as standards. The amount of antigen present in the sample is then determined from the standard curve. Radioimmunoassay is extremely sensitive and specific. It is also least expensive, but it requires special precautions and licensing since radioactive substances are used. This technique was developed by Solomon Berson and Rosalind Sosnin-Yellow at Veterans Administration Hospital, Bronx in New York. For this technique, Yellow received Nobel Prize for Medicine in 1977 along with two other scientists. Procedure. To perform radioimmunoassay, a known quantity of antigen is made radioactive by labeling with gamma radioactive isotope of iodine such as iodine 125 or tritium that remain bound to tyrosine. This radio-labeled antigen is then mixed with a known quantity of antibody. So there will be interaction between radio-labeled antigen and antibody, and that will lead to the formation of antigen-antibody complex. After that, the sample, which can be the serum or any other biological substance, is added to it. So there will be competition between radio-labeled antigen and the unlabeled antigen which is present in the sample for binding with the antibody. As the concentration of unlabeled antigen increases, more of it will bind to antibody, thereby displacing the radio-labeled antigen. So when we add more amount of unlabeled antigen, more of it will bind to the antibody, thereby displacing radio-labeled antigen. As a result, the ratio of antibody-bound radio-labeled antigen to free radio-labeled antigen will decrease. Then the antibody-bound antigens are separated by a suitable method, and the radioactivity of the free antigen that remain in the supernatant is measured by gamma counter. This value is then compared to a standardized calibration curve in order to determine the amount of antigen present in the provided sample. Radioimmunoassay has several applications such as detection of different viral antigens. This technique is also used to detect many hormones and drugs, and also hepatitis B surface antigens and also mycotoxins. Finally, this technique can also be used to detect the early stages of cancer. So friends, this is all about radioimmunoassay. For more updates, please subscribe my YouTube channel, Sam Biology Class.